All right, and we're live again. And I got to unmute you too. And as promised, we're with Dr. Stormy Chamberlain, one of our very esteemed professional advisory board members, and she's going to tell us kind of what's been going on with gene therapy and all of her work. And yeah, so take it away, Stormy.
Today I spent the morning at the Angelman Syndrome Foundation walk. I'm a member of the Designer Gene Club, is what my tattoo says. And I want to challenge the Dup15q group to raise money like the Angelman group did today. I think in Connecticut we raised $18,000. I don't think that's a too lofty goal for the next hour, right? So there's your challenge. Sweet. I know you can do it. I know you can do it. So I'll tell you a little bit about my work. Like I said, I use iPS cells and we convert them to neurons. My lab specifically, I think you heard from James Fink earlier today to tell you, and he probably told you a little bit about how you can use human stem cell derived neurons to learn a little bit about the disorder. But what my lab does is we engineer those stem cells, and we use a fancy tool called CRISPR. You've probably heard of it. In short, the short answer about what CRISPR does is CRISPR just lets us change the sequence of the genome really specifically and really easily. People make such a big deal out of it because it's so easy to use and it's so inexpensive, and it's allowed us to make changes in the genome that weren't previously possible. So how my lab uses it, my favorite way to use it, is we're trying to get rid of the duplicated genes in the Dup15q syndrome. So for instance, we have a cell line that has an interstitial duplication, which means that there are two copies of 15q11 to q13 that are head to tail. And what we're trying to do is get rid of one of those copies. And the reason we want to do this is to make a cell line that is an identical twin to the Dup15q sample we have, but we want it to not have Dup15q. And that way, if we look at the neurons and ask what's wrong with them, we know it's absolutely due to the duplication and not just due to the patient's background, the genetic background that we all have and that differs between all of us. We're also using it to do something called chromosome shattering. This is helpful for the little IDIC. It's cool, right? It sounds good and bad at the same time. What we can do with this is we take CRISPRs and ask them to cut at repetitive sequences. So any sequence that's present a lot of time on a chromosome. So the things that come to mind are where all of those breakpoints are. Many of you know whether your child has used breakpoint 2, breakpoint 3, or breakpoint 4, or breakpoint 5 as part of their duplication. Those are all repeated amongst each other several times. And we've targeted CRISPRs to those repeats. And we've also targeted CRISPRs to a couple of genes, SNORD116 and SNORD115, that we know are repeated in there. And our idea is that we can take, if we treat cells with those CRISPRs, they will preferentially hit that IDIC chromosome because it has so many copies of that repeat. And what we're trying to do is ask the cell to kick that extra chromosome out. And that's what I mean by chromosome shattering, is we think we can shatter that chromosome, and then when the stem cells divide, that one of the cells won't have that little chromosome. And once again, it's a trick to create an identical twin with and without Dup15q syndrome. The other fun things we do with CRISPR, there's actually a CRISPR version that cuts RNA. And so what we're trying to do is take that, it's called, the version we use is called Cas-Rx, and we're trying to use that to knock down multiple genes at the same time. So instead of getting rid of the DNA and getting rid of that extra little chromosome, we can reduce the gene dosage for multiple genes at the same time. And the idea is to start with the 4 or 5 genes that we think are the best candidates to knock down, and see if we can reduce those. Does that fix the cellular phenotypes that we see in the neurons? And then 4 or 5 genes work, can we back it off to 3 genes, or could we back it off to 2 genes, or ultimately 1 gene? So we do it, so we're using Cas-Rx for some of those experiments, although we have some other tricks, antisense oligonucleotides that are actually used as drugs for other disorders. We're using those to do the same thing. Ultimately, though, our big goal is to try to figure out which genes are important for Dup15q syndrome. Of course, we want to do it so that you might be able to design a drug that would reduce those genes. But I think another really cool thing that would be really important for scientists worldwide would be to figure out which genes are important so we can make a better mouse model, and make sure that the mouse models we've made so far, or to make sure that the mouse models we've made so far are the right ones. You may have heard some people talk about using CRISPRs as drugs. And I'd have to say that the field, it's a little bit early to think about that. I think there's a lot of promise for using CRISPRs as drugs, but CRISPRs that cut DNA are not the best drugs. So all of this stuff I told you about getting rid of the duplicons, not a very good therapeutic. It doesn't work very well at all. It's terribly inefficient. And if you leave the CRISPRs in a cell, they will cut DNA that they're not supposed to. And as you might imagine, that's a terrible thing to happen. And if it runs out of its favorite target, it's going to find the next favorite target, and so on and so forth. And again, it's incredibly efficient. So that's not a good thing to have loose in your cells. But that said, I know many people are working on Cas-Rx as a therapeutic. I think we heard from Jean Yao at the scientific symposium last year in Chapel Hill. And he talked about how his lab is using RNA cutting CRISPR versions to do that. And so I think that that's probably a viable therapeutic that people are working on. And once we again understand the genes that are important, that are the most important, we can start tackling Dup15q using that. Wait, one more time? Well, I would punt that to, no, to my knowledge, not that I'm aware of, but again, a neuroscientist such as James Fink may know the answer to that, because they do measure, I know they measured potassium currents. What I can't remember is whether they measured chloride currents or any other. I know he used calcium imaging, and I know they do potassium currents, but I don't know the answer to that. Yeah, wanted to think. Or call Levine at about midnight. I'll give you his cell phone number. It'll be great.
Okay. Can you hear the BP? Okay, got it. Okay, Amy, Chapel Hill, we had our science conference last year in Chapel Hill, and so what Dr. Chamberlain was talking about is Dr. Yao presented some of his data and some of his work there. So that's what she was talking about. But let me know if there's anything else you meant by that. Jen Guns, are we talking about duplications or deletions in the original chromosomes? What about the extra marker chromosome? So, you want to answer that?
Yeah. I mean, so my lab studies both the interstitial duplications and the IDEX, which is the little extra chromosome. And so we're working on strategies. I'll tell you up front that we haven't been very successful yet. And so this is hard. It's, we have to get rid of, 5 to 6 megabases in the case of the interstitial duplications. And in the case of the little IDIC chromosomes, there's like 26 million base pairs on that little chromosome. And we've tried to kick it out before, and we can't get it to leave. It's ridiculously stable. And so we're hoping by the shattering approach it goes away, but we're trying both ways. Again, that's not a therapeutic approach. It's just to give us an important lab tool that I think will be helpful for the whole community. And that gives me a point to say that we also are, I think, very good about sending our cell lines to whomever asks for them. Pharmaceutical companies have asked for them, and we send them out. Individual academic labs have done so. I think we're sending some out to Arnold Kriegstein's lab to study some organoid stuff that they just got a big grant for. So that's the other cool things about resources is we can share.
We definitely, we brought up with Dr. Fink earlier, too. We were talking about when you create cell lines and how sometimes in the field if you have collected them before and there are people that don't always share. And we said we haven't seen that in the Dup15q population at much, but it becomes a problem across developing therapeutics when you can't have access to what's out there. Because he was talking about placing his data after his fellowship in a public source that it can be used and evaluated by other researchers. So yeah, we talked a fair bit about that. That's really important to work with researchers and collaborators that are willing to do that, because when it comes down to it, that's more effective for families and patients who are living with this every day, and it's faster to get to the therapeutics. Cheryl asked, "What's the name of the shadowing method again?" She said shattering.
Shattering. Yes.
Shattering.
Yeah, it was published by two different groups. It was published by two different groups, and one called it Chromosome Shattering. The other one might have had a different name for it, but I forgot. Shattering, I'm visual, so shattering stuck with me. But, essentially it's just using a CRISPR that cuts in a lot of different places, and once you've cut a chromosome enough, it's going to leave. The first time we tried this, we actually got rid of the paternal chromosome 15, so we lost one of the parental chromosomes instead of the IDIC chromosome, but we didn't do enough clones, and so this tells me that it's possible. We just have to be persistent to get rid of that little one, that little isodicentric chromosome that seems to have the glue from hell on it.
That's disheartening to hear. In terms of how much interstitial work have you done?
As of late, we've been focusing more on the interstitial because it's a little bit easier. So in Eric Levine's lab, it's been easier to get phenotypes from the IDIC cells. And in fact, all of James Fink's work on that is publicly available at bioRxiv. It's a preprint, so it hasn't been peer reviewed, but again, it's publicly available, so anybody can look at that data. So we've had better luck getting phenotypes from those cells, but Eric's lab has been looking at the interstitial duplications a little bit more as of late because they're a milder form, and we're wondering, are there the same phenotypes that you would see in IDIC, only less strongly affected? Or are they very different phenotypes altogether, and are we really maybe dealing with two different disorders physiologically? And my lab has been focused on the interstitial duplications because they're a little bit easier. To get rid of the extra duplicon, we've inserted LOX-P sites, and in fact we have successful clones that have the LOX-P sites in there, and now we're using a drug trick, negative selection, to get recombination between the two LOX-P sites and ask one of the copies to leave. So we've been focusing a little bit more on the interstitials lately, but all of our past work has been on IDICs.
Cheryl is asking, can you summarize the difference between the two methods? I think she's talking about between chromosome shattering and CRISPR. I think that's what she's talking about. Cheryl, correct me if I'm wrong, but I think that's what she's asking about. Just a summary of how they're different.
So they're just different. Let me think of a good analogy here. They both use CRISPR. We're both using the fact that we can cut the genome, and by making a cut, we can more efficiently change the genome. But the strategy is very different between the two. In one case, we stick in these little sites called LOXP, and it mediates recombination, so they match up with each other and cause the copy to leave. Whereas the shattering, the idea is it's actually really crude. It's just to break up that chromosome enough that it can't stick around, that it gets lost when the cell divides. So conceptually, they're very different, but we use the same types of CRISPRs to do both.
Okay. Jen, so if the original chromosomes are normal and the extra marker chromosomes are the issue, you risk deleting the parental chromosomes with this technique? Question mark.
Absolutely. Yep. And so what we have to do is we take a bunch of cell lines that we know have seen the CRISPRs, and then we ask, do they have mom's chromosome, do they have dad's chromosome, or do they have the IDIC chromosome? And we can follow this using DNA methylation. So it's a lot of work, and the students who take this on are brave or stupid, I'm not sure which. There's a fine line between the two, but nothing ventured, nothing gained, right?
Yeah. We were talking to James earlier about how fast science is changing, and how he started his work and how slow it was, and now when you move 4 years down the road, how much quicker you can do exactly the same work. And I said, like, does it make you feel like this? He said, no, it makes me feel. He always says he's so positive. He's like, no, it makes me feel energized. I'm like, yeah, I don't think that's always what the case is.
I sometimes look at this and think, oh my goodness, we have such a long slog ahead of us. So, many of you know I work in the Angelman field as well, and that's a single gene disorder, right? And, they really are getting close to some therapeutics, but they have one gene, and there's multiple ways to put that gene back. And so I keep thinking about Dup15q, and I'm like, oh man, it's such a long slog, but things advance quickly. And once we get an idea, all of the tricks that they've used for single gene disorders all of a sudden apply to us. And so our goal is to, and thank God I'm simple-minded, because if we can get this down to like just a couple of genes, I think we can take off, and then it starts to become really fast, at least a couple of approaches. And meanwhile, you have all these brilliant neuroscientists like James, and also pharmaceutical companies, I think like Roche and Takeda and some of these others that will say, oh, look, well, we think that this might be amiss based on the physiology of cells. And that's awesome, because they can do that too. And I think having multiple shots on goal is a great way to go in terms of therapeutics.
My question is, so say you are successful with these the tough cells that don't want to break up, and they're just stuck there. Say you're successful with that. What does that mean? What's the next step? So you've broken it up and you've kicked it out. Now what?
Yeah. So again, I like to hedge my bets a little bit. So one of the other things we've been working on is we developed antisense oligonucleotides to several of the genes, and we've had them for a while, and we know they work, and we kind of know how to dose them. But and I'll tell you, I hope Eric doesn't mind me giving away all his secrets. But one of the things that he has a student doing is taking the I hope I get this right. She's taking the interstitial duplication cells and knocking down UBE3A in those, because we think UBE3A is probably a major player. So she's knocking down UBE3A and asking, does that restore the physiology? And her only problem is she really doesn't have a typical cell line to compare it to. And so we know whether it can change the physiology of the Dup15q cell, but we don't know whether it's made it like a typical cell again. And so that's why we really need to get that typical cell. But nonetheless, she's doing it anyhow. And the other thing she's doing, I think this is kind of clever, she's taking paternal interstitial duplications cells. So they don't have the disorder, but they do have the duplication on dad's copy. And now she's trying to, now this is going to be a little confusing, but we have antisense oligos that knock down the antisense transcript to UBE3A. So she's actually upregulating UBE3A in those cells to ask, if we take a cell that we know is not affected with Dup15q syndrome and we upregulate UBE3A, does it now have the same features as our cells with Dup15q syndrome?
So so one kind of response or reaction to that is, though, I know that in the past the paternal duplications were kind of set aside as something special, that they didn't initially exhibit the syndrome. But now, as we're finding more of them, and we're actually phenotyping them, that they have a phenotype. It might not be Dup15q syndrome phenotype. So a lot of it comes, I know there's a lot of parents listening to this. We talk a lot about this because when you read the genetic report, they don't have methylation assays most of the time. And so when we let them in, when we talk about paternals and maternals, the duplications, and we talk about it a lot. We're trying to address it on the board, how to make it more clear for families. But that it's not necessarily the same syndrome, but there's a phenotype that's associated with it. Are you seeing a different phenotype in those cells, though? Like they're not showing the syndrome phenotype, but there's a different phenotype from control.
Yeah. So the dirty little secret is that our phenotypes are quite crude. And thus far, we haven't seen any phenotypes. We've tried to look at them in many different ways. We've tried to, James is, when he was a PhD student, he looked at, how many signals do the neurons send? How are they receiving signals? He's asking like whether they have homeostasis. Do they come back? Do they adjust appropriately to the signals they're getting from outside and things like that? So he's asked a lot of questions of them. And so far, the paternal interstitial dupes they're not different from the typical cells that we have so far. So, but that said, I'm sure we haven't looked at everything.
And you're looking at the same sort of thing with the firing, in terms of the spiking.
Right. And so that's what they would look at in Eric's lab. And then in my lab, we just try to develop these tools so that they can apply it to physiology, right? So we can make the antisense oligos again to knock down UBE3A and restore its level, so they can say, well, does that work? We can make a cDNA clone. We can put the DNA back into the cell that has UBE3A, right? So we can overexpress only UBE3A and ask, does that also give you a Dup15q cellular phenotype or not? And then so we've made also antisense oligos to the GABRB3 gene. What's the other one we have? HERK2. The one that still gets us is GABRA alpha 5, GABRA5. So we've made a CRISPR for that. When it's low on my totem pole because it's harder to hit, but we keep making tools so that Eric and his lab can test them and see if that's the gene.
Yeah, that's great for us. Are the marker chromosomes harder to break up because of the mirrored replication in the maternal marker? I think that's what she meant.
Yep. Yeah. So I don't know why they're so stable, but I think you're right. I think it's because they're mirrored, and there's two centromeres, and many of us scientists would have thought, ooh, well, that's terrible. That should destabilize them, but apparently not. So I don't know if it's their small size or the two centromeres, but they certainly seem to be very stable. They're certainly not lost spontaneously at any decent frequency. We've tried that one already.
So, just speaking for myself, as someone who understood about 10% of what you just said, that's okay. No, I understood a little bit. I can follow, but I can't ever spit it back out. How can we participate? How can we help push this forward? Like, do you need samples? Do you need participation? Are there studies? Like, how can parents who are willing and able help you?
Yeah. So every now and then we need samples, but it's gotten to the point now where we're after really specific samples. People who are mosaics who have some typical cells and some IDIC cells. Those are particularly valuable to us because that's another way in which we can get genetically identical twins with and without Dup15q syndrome, which is really what we're after.
Can you do
but other than that,
can you explain mosaicism? We do talk about it a lot, but can you just give the 20,000 foot version of mosaicism?
Sure. Yeah, mosaicism means that really early on in the development of a baby, some of the cells have the extra chromosome. In the case of IDICs, I'll just boil it down in the case of IDICs, some of the cells have the IDIC chromosome and some of them don't. And this happens early on through ways that I don't always understand. It could be that the cells lost the IDIC chromosome, although I'm finding that harder to believe. Or it could mean that the duplication didn't finish until after fertilization happened. At any rate, the result is a human being that has some cells with Dup15q and some cells without. And they're all mixed through the body. They're if I could grab my cat here, who's not a calico, you would sort of, calico cats, they'll have some spots. For instance, they might have orange in the middle and black here, right? But not every calico cat looks the same. And that's mosaicism. That cat is mosaic for orange and black. And so just like that, so there are some humans who are mosaic for Dup15q and might have the IDIC chromosome in some cells and not in other cells. And so if we took a blood sample from that person, there's a good chance that some of the blood cells would have the IDIC chromosome and some would not. And we could isolate stem cells from both the typical cells and the IDIC cells.
So that's what you mean in terms of the creating identical twins that are not identical twins that have Dup15q, don't have Dup15q. You're creating stem cells from the mosaic positive cells and the mosaic negative cells.
Yeah, and they would be identical twins. They would be identical twins except for that little chromosome.
Mm-hmm. We do have a handful of mosaics as well, IDIC mosaics.
Yeah, and so like samples, like those samples right now would be really, really helpful. And if you could pass some of those to me, and we could capture the cells, that would be a great help to what we do. And the other thing I think you guys can do to help is really support the research. That I think that's what the big give is about. Supporting the research because the things that Dup15q Alliance will fund are pilot studies that help us prove the science is good, and then we can ask for funding from larger agencies like the Simons Foundation and the NIH. That's kind of, I think, the model that works really well is you fund some stuff that's a little bit risky, but high risk, high reward. And then we can take it to the NIH and once it's a little bit de-risked, and we have some good preliminary data, it makes it a lot easier for them to fund us.
In the same token, that in terms of what the Alliance does and brings, and if you're a research funding, a research program is, all families have the same questions. When they come in, they have the same questions about inheritance and phenotype and that sort of thing. And without funding research and honestly, researchers aren't, most of the time they're not going to do it out of the kindness of their heart. They might do it out of an interest, but at the end of the day, they need to feed their families too. So they need to have some sort of funding that comes along with it. So these questions can't be answered without the research that they're doing, and they're dedicated to Dup15q. Our small pilot grants, they're a lot of money when it comes down to a small organization, but they can really grow. Like that investment of $1 that then turns into $10 when it goes to an NIH grant because it's pilot data is really what our researchers need to keep moving that ball forward. So in terms of sharing these things and investing and giving a donation monthly or any donation you possibly can, or just telling somebody, instead of giving we don't, for Christmas this year, can we do donations to Dup15q because we want to invest in the research that they're doing. It's really important. I know that I do it for my kids because one, I'm so sick of cleaning up toys, and two, it makes people feel good to be able to give to a larger, something bigger than themselves. So it's definitely something like all of these questions that are coming in, they're so important and so interesting, and families really want to understand them. Without the research that the Stormys of the world and the James Finks of the world and the Eric Levines of the world are doing, we're not going to know those answers. And it will just be talking about comparing and contrasting kids and how they look clinically and not really getting down to the nuts and bolts of what we really need to know.
Well, and to be honest, pharmaceutical companies become interested based on academic research too. They also don't like to do risky things. And so they kind of wait for an important and tractable finding from an academic lab, and then they'll invest in it. So that's kind of the best way to get pharmaceutical companies interested as well. And a case in point, Dup15q Alliance funded James Fink for a couple of years, right? And based on the work that he did, Eric and I earned a almost $200,000 award from the NIH to study the interstitial duplications. And you know, it's $200,000, but actually the other thing we have to pay for is, you know, to keep the lights on at our buildings. And so those are monies that come to our university. And we don't charge, like Dup15q Alliance doesn't pay a huge amount of those, but the NIH pays a substantial amount to help keep our universities open. And so really it's $200,000 to Eric and I to do work, but it's like $400,000, $500,000 to our university to help keep the lights on and things like that. So
F&A, literally F&A. I'm not being F&A. It's one of those things. I mean, I did a slide in 2017. I'll do another one that the growth that we see, like Dr. Jeste also talked about the work that she's been doing between the IDDRC. We did fund her for a grant 4 years ago. That was a small amount that got her pilot data to get the IDDRC grant, and then now she made it into an R13, so we're getting federal money back. And again, so that's just an investment that we make. We make it with under the advisement of a lot of scientists. So our grant cycle just closed yesterday. We got all of our applications. Actually, we have one that's a straggler who let me know they're a straggler. But we have those grants coming in, and they're going to be evaluated by a group of external scientists that can say this has merit, this can move things forward, and this is a good high-risk, high-reward investment for the Alliance that's going to create more interest and more funding. So it's something like that. And we do need to keep our universities open. We need to keep Stormy in a job, because if she doesn't bring in that facilities and administration cost from the NIH, they're going to turn the lights off. We don't need that to happen. So.
Well, more importantly, we need to pay the people that do the work. You have to spend a lot of time writing grants and writing papers and teaching, and it's actually our students who do the work. And so, I was at the Angelman Walk today, and it's kind of a bit of a lab reunion. And what's really fun is to see my students who've been flown the coop a couple of years ago come back, and they still participate in the disorders that we study, and they take that knowledge with them where they go. One of my students is at the Translational Neuroscience Core at Boston Children's Hospital, and the other one works for Novartis in their neurodevelopmental drug discovery program. So we educate students that then take Dup15q out into the world, and at some point may very well bring new thoughts to it and new efforts. The cat doesn't help at all.
Other than comedic enjoyment.
Yeah.
Questions, ladies?
No, I think the main point to hit is that just a little bit of funding can create more funding and more funding that we don't necessarily have to raise. We just have to get you there, and then you can make that apply for bigger grants and things like that. So just support from the community to keep this going.
Absolutely.
And then also
keep our chin up every now and then when we get discouraged. I know you guys need encouragement and support, and so do we.
Well, the other thing is when we ask, when we send out emails about the research that's going on, human-based or otherwise, or when our genetic counselor intern reaches out for more extensive genetic reporting, the reason we're doing that is to work with pharma and work with industry and work with researchers, because they'll let us know we need to find 3 more mosaics, eidetic mosaics. Can we find those? And we say, yeah, we have them. and we know that they have it, and because they showed us their genetic report, or something to that effect. And based on something we said before, we keep all of that, under lock and key. It's all, under our IRB and that sort of thing. But it's for a reason that we're asking. It's not just because we really want you to have to go through that binder and find it. It's because people are asking for it. They're asking for it for a reason. They're asking for scientific missions, and we're being asked for it a lot more. Put it that way. Even in, it's definitely something to hear more about.
Well, and you'll be glad to have that. The other reason to participate in studies like that and to join the registries and things like that is you have to do a lot of this legwork before any clinical trials can happen. And if you wait until a clinical trial is ready, it's too late. That means that's going to delay the clinical trial. And so as again, the Angelman folks are finding that having natural history studies and clinical data collection and genetic reports of patients is really helpful to help pharmaceutical companies, first of all, be willing to move forward, but second of all, they have to have that information so they can compare treated and untreated. That's ultimately what they need to do.
I think it just goes back to what Luke was saying earlier, is in order to get attention from anyone, we need our community to come together. I mean, we really have come together. But to stay organized and pushing forward so that we keep getting more researchers who are interested and more funding towards pushing for epilepsy and autism and adjusting the extra duplications.
Yeah. Luke was on here. Luke was on here representing KIF1A.
Okay.
Yeah.
Okay.
Yeah.
I didn't know whether he was representing KIF1A or Ovid, right?
Yeah. No, he was on there representing KIF1A. We were talking about advocacy and just his relentless spirit, because that word will always go with him now.
Yeah. It takes that.
He does. Yes. Yeah. So we're talking about Ovid later, but he was representing KIF1A. So yeah. I was connected with Vanessa earlier this week through I'm going to get this wrong, Polygenic Gene Consortium. Ed is really, we got to call it multigenic. They have not decided what the proper term is, and I don't think anybody knows. So we're just going to make something up and pretend that that's what it means. So it's something like that. Lots of genes.
Yeah, but I think it's important. It's important to think bigger too in terms of connecting, and the Dup15q Alliance has always been nicely connected to the Angelman Syndrome Foundation, and it's great to learn from both organizations, to learn from each other. But I think like this Polygenic Consortium is also going to be really helpful. First of all, learning from each other, but also it's the way we give to others from our experience, and I think that's great for the community as a whole. As a scientist, it challenges me to come up with cool ways to use our tools.
I'll talk a little bit more about I haven't given my presentation yet, so I'm going to talk about that in the collaborations in a little bit too. So I make sure remind me to bring that up. But yeah, Ed and Brenda, like, are you sure you're using the right word? I'm like, we were going back and forth about what the right word is. Like, we still haven't decided on it, but nothing's trademarked yet. So.
You can use the bad and confusing term that they use in the Prader-Willi land, and that's contiguous gene syndrome, which sounds terrible, right?
That sounds awful. That's almost like shattering chromosomes.
I know. Not nice.
All right. Well, thank you so much for all you do for us. And I mean, you're always a person that I oftentimes call and text with very random questions, and out of the blue. But thank you so much for all you do for our organization, and also Angelman. I know that collaboration is something that we really, really treasure and will for a very long time. So thank you.
Oh, thank you guys. My pleasure. If you find any questions later, hook me up. Happy to answer.
So this is going to go live on our so it'll be on our Facebook, and anything that comes up later, any questions or anything like that we didn't read, it'll be up there. We'll send it to you to answer.
Okay.
So.
Cool.
All right. Thanks. Have a great evening, and enjoy that beautiful weather.
Yeah, thank you. You guys too. Happy weekend.
All right,
Bye.
Bye.
All right. So.
Cool. I want to listen to that.
Let's go back to this. So, all right, that was our very own Stormy Chamberlain. it looks like a very nice back deck.
Mm-hmm.
I want to go sit in the trees. What's up next?
So it was going to be Luke, so we can
talk about this.
You can do your thing.
Okay.
Or we can do it and then-
Right. So we're going to turn this live off and start a new one. And the next one will be actually me. I'm going to give you a little bit of update about what I've been up to, where I've been traveling. These ladies are going to help me remember where I've been traveling by interviewing me a little bit. I do have my schedule, so I can, kind of the outcomes. I do try to make the Facebook post, but just the outcomes of all of those travels. And if you have any questions for me, I will be watching them and answering them real time. I will answer anything from the organization side of things to what do I do, and I don't know, when do I sleep at all, which I usually don't. So we'll be back in a second. I'm going to end this one and we'll start another one. Sound good?